Dr. Vijay Kumar Singh, and today I will be discussing with you all about an interesting and learning episode of B.Pharma on the unit of antiviral agents, which is too important unit of medicinal chemistry 7th semester. Let's start our episode related to module 1, 2, 3 and 4. While taking a look at what we are going to learn today in this regard, module 1, Introduction to Viral Disease. Viruses are microscopic infective agents that may lead to mild to severe illnesses in humans, animals and plants. The virus particles are 100 times smaller than a single bacterial cell. The bacterial cell alone is more than 10 times smaller than a human cell, and a human cell is 10 times smaller than a diameter of a single human hair. Viruses by themselves are not alive. They cannot grow or multiply on their own and need to enter a human or animal cell and take over the cell to help their multiply. These viruses may also infect bacterial cells. The virus particles or the virions attack the cell and take over its machinery to carry out their own life processes of multiplication and growth. An infected cells will produce viral particle instead of its usual products. The structure of virus, the free living virus particles is termed as virion and consists of three main parts. Nucleic acid, this is the core of the virus with the DNA or RNA, that is the deoxyribonucleic acid and deoxyribonucleic acid respectively. The DNA or RNA holds all of the information for the virus and that make it unique and helps it multiply. Protein coat, capsid, this is a covering over the nucleic acid that protect it. Lipid membrane, envelope, this cover the capsid. Many viruses do not have this envelope and are called naked viruses. Fundamentally, viruses are classified into two main groups. First one is deoxyribonucleic acid DNA viruses, ribonucleic acid RNA viruses. Module 2, virus life cycles. Replication of DNA viruses. DNA virus enters the host cell nucleus where the RNA polymerase catalyze the mRNA transcription and translate of mRNA into virus specific protein take place. Some of these proteins are enzymes that synthesize more viral DNA as well as viral coat and envelope. After coating around the viral DNA, complete virion are released after host cell lysis. Replication of RNA viruses. Enzyme presents in virion synthesis its mRNA from viral RNA template. Sometimes viral RNA act as its own mRNA. This is translated by the host cell into virus enzyme including RNA polymerase, which directs the synthesis of more viral RNA. Assembly and release of virions occurs same as in DNA virus. The viral life cycles may outline under: first, the virus is going to attach to the host cell, then adsorb to the cell surface where it invades the host cell membrane, then viral particle is encapsulated by the host cell. Then penetration is going to occur. After penetration, there is a process that is called uncoating, where the viral DNA and RNA is passes into the host cell. After that, the RNA and DNA virus is going to occur. After that, the transcription is going to occur. After the transcription, synthesis and binding of the viral mRNA to the host cell ribosome. After that, after the completion of viral mRNA synthesis, the synthesis of viral DNA oblique RNA and capsid protein is going to be occur. After encapsulation, the mature virion is going to be released after the host cell lysis. Viral diseases. The types and symptoms of disease produced depend on the group and species of virus. Many diseases are produced by the different group of viruses. DNA containing virus, adenovirus, it have many types and the disease occurs by this is respiratory tract and eye infection, that is known as the conjunctivitis. Second one is herpes virus. It is a herpes simplex virus. It causes the eye infection and skin diseases. Pox virus, variola vacciana. The disease caused by this is varicella. It is also called as chicken pox. Next category of virus is RNA containing virus. The examples are orthomyxovirus. It causes influenza A, B and C. Next is paramyxovirus. It causes mumps, measles and parainfluenza. Next is picronavirus. It causes respiratory disease, gastrointestinal disease and poliomyelitis. Next category is retrovirus. It is a C-type virus. It causes leukemia and lymphoma. Next is toga virus. It is a rubella virus and it causes hepatitis agents. Ideally, a drug is considered effective if it interferes with replication process without affecting the normal cellular metabolic process, nucleic acid and protein synthesis of the host. Unfortunately, this target has not been achieved by large number of antiviral compounds which have been tried in viral infections. Most of them were successful at therapeutic level, but due to their toxicity and some were not effective enough against variety of viral infections. In most of the viral infections, immunity of the host cell at the time of infection is not well understood. The result is that the symptoms of viral infection may not appear until viral replication process are complete, and due to this latent period of absence of specific symptoms, it is difficult to determine the effectiveness of an antiviral drugs. Several antiviral agents have been discovered that block specific virus coded enzymes which produced in the host cell and essential for viral replication, but not for the normal host cell metabolism. This chapter is mainly concerned with antiviral agents that have been approved by United States Food and Drug Administration US FDA and are clinically effective in viral infection. The antiviral agents are classified according to their specific mode of action and possible interruption of the viral replication by these agents. Agents act on early stages of viral replication. Amantadine hydrochloride. Amantadine hydrochloride is a symmetrical tricyclic amine that inhibits penetration of virus particle into the host cell. It also inhibits the early stage of viral replication by blocking the uncoating of the viral genomes and transferring nucleic acid into the host cell. Clinically, it is very effective in preventing and treating all strains of influenza A. It also shows its in vitro activity against influenza B and some RNA viruses. Generally, the drug has low toxicity at therapeutic levels, but severe central nervous system symptoms such as nervousness, confusion, headache, drowsiness, insomnia and coma occurs at high dose. Its therapeutic usefulness, however, appear to be limited. This drug has been used continuously for Parkinson's disease for many years. Rimentadine hydrochloride. Rimentadine hydrochloride is alpha methyl 1 adamantane methyl amine hydrochloride. It appears effective in clinical trials. This drug has action similar to those of amantadine, but 4 to 10 times more active. However, the CNS toxicity occurs less often than the amantadine. Interferon. Interferon are cellular glycoproteins produced naturally by virus infected cells and have protective action against viruses. Its displayed biologic properties including inhibition of viral growth, inhibition of cell multiplication and immunomodulatory activities. They are not virus specific and can interfere with a wide variety of DNA and RNA viruses. They are therefore considered as potent broad spectrum antiviral compounds with a high therapeutic ratio. There are three classes of interferons termed as interferon alpha, interferon beta and interferon gamma. Interferon alpha is not a single substance but a family of approximately 20 proteins with similar activity. Interferon alpha and interferon beta are produced by B and T lymphocytes, macrophages and fibroblasts in response to the presence of virus and cytokines. They have 166 amino acids. Interferon alpha and beta have antiviral activity and interferon alpha also have anti-tumor activity against some lymphomas and solid tumors. Interferon gamma termed immune interferon and produced mainly by T lymphocytes. It have 146 amino acids and more immunomodulators than antiviral activity. Interferon act by production of enzymes that inhibit the translation of mRNA into viral proteins, thus inhibit viral replication. Module 3.2 The agents which interfering with nucleic acid replication. Acyclovir. The era of effective antiviral therapy begins with acyclovir. This is a synthetic analog of deoxyguanosine in which the carbohydrate moiety is acyclic. This agent is active against certain herpes virus infection. This virus induces virus specific thymine kinase and DNA polymerase. Acyclovir inhibits this enzyme. Thus, acyclovir significantly reduces DNA synthesis in virus infected cells without disturbing the active replication of uninfected cell. Acyclovir is radially converted into monophosphate within the cell by thymidine kinase. This reaction occurs much faster by infected cells than by the normal cells because acyclovir is a poor substrate for the normal cell thymine kinase. Acyclovir monophosphate further converted to di and triphosphates. It is the acyclovir triphosphate that inhibit viral DNA polymerase. Terminating the nucleotide chain, it is 30 times more potent against herpes virus enzyme than the host enzyme. Idoxuridine. Idoxuridine is a nucleoside containing halogenated pyrimidine which is structurally related to thymidine and act by competing with it in the synthesis of DNA, thereby preventing the utilization of thymidine. The drug interferes with the synthesis of normal cellular DNA and hence it is too toxic for systematic use. It has been employed locally as 0.5% drop or ointment in the treatment of herpetic keratitis. 5-iodo-2-deoxyuridine. Trifluoridine. Trifluorothymidine is a fluorinated pyridine nucleoside structurally related to idoxuridine. It is a potent specific inhibitor of replication of HSV1 in vitro. Like other anti-herpes drugs, it is the first phosphorylated by thymidine kinase to mono, di and triphosphate form, which are then incorporated into viral DNA instead of thymidine to prevent the formation of viral mRNA and synthesis of virion protein. Cidofovir. Cidofovir is a synthetic acyclic pyrimidine nucleotide analogous of cytosine. It undergoes phosphorylation by host cell enzyme to its active intracellular metabolite cidofovir diphosphate. It has antiviral effect by interfering with DNA synthesis and inhibiting viral replication. Kytrabin. Kytrabin is a pyrimidine nucleoside related to idoxuridine. It acts by blocking the utilization of deoxycytidine, thereby inhibiting the replication of viral DNA. The drug is first converted to mono, then di and triphosphate, which interfere with DNA synthesis by inhibiting both the DNA polymerase and reductase that convert cytidine diphosphate into its deoxy derivative. Famiclovir. Famiclovir is a synthetic purine nucleoside analog related to guanine. It is the diacetyl 6-deoxy ester. It is a prodrug of panciclovir, which is structurally related to ganciclovir. Pharmacologically, it is similar to acyclovir that inhibit viral DNA polymerase. Formivirsin. Formivirsin is the first antisense oligonucleotide agent that has been approved as an alternative medicine for patients with CMV retinitis for whom other agent did not work. Phoscarnet. Phoscarnet sodium is the trisodium phosphoformate hexahydrate that inhibit DNA polymerase of herpes viruses. It is not phosphorylated into an active form by the host cell enzymes. Therefore, it has the advantages of not requiring an activation step before attacking the target viral enzyme. Ganciclovir. Ganciclovir sodium is an acyclic deoxyguanosine analog. Ganciclovir inhibits DNA polymerase. Its active form is triphosphate, which is an inhibitor of viral rather than of cellular DNA polymerase. The mechanism of action is similar to that of acyclovir. However, it is more toxic than acyclovir to human cell. Vidaravin. Vidaravin is an adenosine nucleoside obtained from culture of Streptomyces antibiotics. Cellular enzyme convert vidaravin to mono, di and triphosphate derivatives that interfere with viral nucleic acid replication, specifically inhibiting the early step of DNA synthesis. This is a three-step process in which vidaravin is sequentially phosphorylated by kinases to triphosphate ARA ATP. This prevents the reduction of nucleotide diphosphatase, causing a reduction of viral replication. Ribavirin. Ribavirin is a guanosine analog used to stop viral RNA synthesis and viral mRNA capping. Thus, it is a nucleoside inhibitor. Ribavirin is a prodrug which, when metabolized, resembles purine RNA nucleotides. In this form, it interferes with RNA metabolism required for viral replication. Multiple mechanisms may be responsible for its action against RNA viruses. It is incorporated into RNA as a base analog of either adenine or guanine. It pairs equally well with either uracil or cytosine, inducing mutation in RNA dependent replication in RNA viruses. Such hypermutation can be lethal to RNA viruses. Against DNA viruses, it effect on mainly DNA viruses, which suggests that the drug functions only as an RNA nucleosides mimic and never a DNA nucleoside mimic. Ribavirin 5 monophosphate inhibits cellular inosine monophosphate dehydrogenase, thereby deleting intracellular poles of GTP. List of miscellaneous classes of antivirals. Antiretroviral agent including protease inhibitor, nucleoside reverse transcriptase inhibitors. Zidovudine. Zidovudine is an analog of thymidine in which the azido group is substituted at 3 carbon atom of dideoxyribose moiety. It is active against RNA tumor viruses that are causative agent of AIDS and T cell leukemia. Zidovudine is converted to 5-mono-di and triphosphate by cellular enzyme. These phosphates are then incorporated into proviral DNA, resulting in termination of DNA chain elongation. Because of presence of azido group in zidovudine, diadenosine. Diadenosine is a purine dideoxynucleoside which is analogous of inosine. It is a prodrug that is bioactivated by metabolism of dideoxyadenosine triphosphate, which is competitive inhibitor of viral reverse transcriptase and is incorporated into the developing viral instead of deoxyadenosine triphosphate. It inhibits HIV reverse transcriptase and exert virus static effect on retrovirus. Delcitabine. Delcitabine is a homologue of cytosine. It is a useful alternate drug to zidovudine and it is given in combination with zidovudine when CD4 cell count fall to less than 300 cell per millimeter cube. Lamivudine. Lamivudine is 2-3-dideoxy-3-thiacytidine and commonly called 3-TC. It is an antiretroviral drug used to prevent and treat HIV/AIDS and also used to treat chronic hepatitis B. It is of the nucleoside analog reverse transcriptase inhibitor. Lamivudine. It is analogous of cytidine. It can be inhibit both type 1 and type 2 HIV reverse transcriptase and also the reverse transcriptase of hepatitis B virus. It is phosphorylated to active metabolite that compete for incorporation into viral DNA. They inhibit the HIV reverse transcriptase enzyme competitively and act as a chain terminator of DNA synthesis and therefore, the viral DNA growth is terminated. Abacavir. It is also called as ABC. It is an antiretroviral drug used to prevent and treat HIV or AIDS. It is a nucleosides analog reverse transcriptase inhibitor. Viral strain that are resistant to zidovudine or lamivudine are generally but not always sensitive to abacavir. It is a well tolerated. The main side effect is hypersensitivity. In this drug, the carbocyclic ring is attached to the base instead of sugar. It is available in combination formulation abacavir, lamivudine and zidovudine, abacavir, dolutegravir and amlodipine and abacavir amlodipine. ABC is an analog of guanosine, a purine. Its target is the viral reverse transcriptase enzyme. After ABC is completely phosphorylated, it competes with guanosine for incorporation into the DNA strand. When the reverse transcriptase incorporates the ABC product, transcription is terminated. Emtricitabine. Emtricitabine is an oral active nucleoside reverse transcriptase inhibitor. It is enantiomer is more active than isomer. Stavudine. Stavudine is a pyrimidine nucleoside analogous that have a significant activity against HIV-1 after cellular conversion of the drug to D4-T triphosphate non-nucleoside reverse transcriptase inhibitor. Nevirapine. Nevirapine is a dipyridodiazepinone derivative that bind directly to reverse transcriptase and inhibit it. Its block RNA and DNA dependent polymerase activity by causing disruption of catalytic site of enzyme. Delaviridine. Delaviridine is a biheteroaryl piperazine derivative. It is a potent non-nucleoside reverse transcriptase inhibitor activity for HIV-1. It directly inhibits reverse transcriptase and DNA directed DNA polymerase activity of HIV-1. Efavirenz is used as a part of highly active antiretroviral therapy for the treatment of a human immunodeficiency virus type 1. Efavirenz is also used in combination with other antiretroviral agents as a part of an expanded post-exposure prophylaxis regimen to reduce the risk of HIV infection in people exposed to a significant risk. Example, needle stick injury, certain type of unprotected sex, etc. Efavirenz was combined with the HIV medication tenofovir and emtricitabine, all of which are reverse transcriptase inhibitor. It's come in NNRTI classes of antiretrovirals. Both nucleoside and non-nucleoside reverse transcriptase inhibitor have the same target. The reverse transcriptase enzyme, an essential viral enzyme which transcribes viral RNA into DNA. Unlike nucleoside, RTIs which bind at the enzyme active site, NNRTIs act allosterically by binding to a distinct site away from the active site, known as NNRTI pocket. Efavirenz is not effective against HIV-2 as the pocket of HIV-2 reverse transcriptase has a different structure which confer intrinsic resistance to the NNRTI classes. Efavirenz is a potent inhibitor of wild type as well as resistance mutant HIV-1. Methicillin. Methicillin is an antiviral drug that works by interferes with the translation of mRNA messages into protein synthesis on the cell ribosome, producing a defect in protein incorporation into virus. Active against pox viruses including variola and vaccinia. It is also used as a prophylactic agent against smallpox. Some newer antiviral drugs. Emtricitabine. Emtricitabine is a nucleoside reverse transcriptase inhibitor for the treatment of HIV infection. It is marketed in a fixed dose combination with tenofovir. A fixed dose triple combination of emtricitabine, tenofovir and efavirenz is marketed under the brand name of Atripla and approved by US FDA in July 12, 2006. Emtricitabine is cited in analogues and work by inhibiting reverse transcriptase, the enzyme that copy HIV RNA into new viral DNA. By interfering with this process, emtricitabine can help to lower the amount of HIV or viral load in host cell and can indirectly increase the number of immune system cells, that is called T cells or CD4 T cell. Tenofovir. Tenofovir is used to treat HIV or AIDS while it is a prodrug. Its active substance is tenofovir. This drug is selectively interfere with the action of reverse transcriptase and weakly interfere with the host cell DNA polymerase. It prevents the formation of the 5 to 3 phosphodiesterase linkage essential for DNA chain elongation. Once incorporated into a growing DNA strand, It causes premature termination of DNA transcription. Itaravirine. Itaravirine is a drug used for the treatment of HIV. It is a non-nucleoside reverse transcriptase inhibitor. Unlike the currently available agent, that resistance to other NNRTIS does not seem to confer resistance to itaravirine. In January 2008, it was approved for the use by US FDA. Itaravirine is a chemically diaryl pyrimidine having some conformational isomerism that can bind with reverse transcriptase enzyme even in the presence of mutations. Many diaryl pyrimidine analogues are currently being developed as a potential anti-HIV agent, notably Rilpivirine. Itaravirine is a second generation NNRTI designed to be active against HIV with mutations. Darunavir. Darunavir is a protease inhibitor used to treat HIV infection. It was approved by US FDA on June 23, 2016. Darunavir is a second generation protease inhibitor designed specifically to overcome problem associated with the other existing protease inhibitor agent. Early protease inhibitor often have susceptibility to drug resistant mutations, severe side effect and required a high therapeutic dose. Darunavir represent a treatment options for the people with drug resistant HIV. Raltegravir. Raltegravir is an antiretroviral drug used to treat HIV infection. It received approval by US FDA on 12 October 2007 as a new class of HIV drug. Raltegravir targets integrase, an HIV enzyme that integrates the viral genetic material into human chromosomes, a critical step in the pathogenesis of HIV. The drug is metabolized away by glucuronidation. Raltegravir is available in chewable pills which are only approved for use in children 2 to 11. Older adolescents will use the adult formulation. Maraviroc. Maraviroc is an antiretroviral drug used in the treatment of HIV infection. It is classed as an entry inhibitor. Specifically, Maraviroc is a negative allosteric modulator of the CCR5 receptor, which is found on the surface of certain human cells. The CCR5 is an essential co-receptor for most HIV strains and necessary for entry process of the virus into the host cell. The drug binds to CCR5, thereby blocking the HIV protein GP120 from associating with the receptor. HIV is then unable to enter into the human macrophages and T cells. Module 4. Conclusion. After going through the manuscript, we can conclude that from the beginning to till date, availability of antiviral against viral infection has increased several fold. New strategies based on the growing understanding of the biology of pathogenesis viruses and their action and in the host cell could well, if vigorously implemented, have potential to target the viruses causing most viral disease. However, the ultimate weapon in the fight against the virus is vaccination. This has been proved to be stunningly effective against diseases such as polio and smallpox, and more recently against influenza and hepatitis B. However, there have been many clinical trials. The prospect of vaccine against HIV still seems rather remote. Now, we have understood the module 1, 2, 3 and 4. With all this information, here we come to end of today's lecture. This lecture will be very helpful for you. Study hard and enhance your knowledge. Time for you to self-study. All the best. This is Dr. Vijay Singh signing off. If you want to learn more and enhance your knowledge, you may log on to our website for MCQ, quizzes and LOR. Website is www.cec.nic.in. Till then, keeping studying. Goodbye.